Welcome back. Today we're going to talk about enzymes, the cell membrane a little bit more, and the types of membrane transport. All right, so let's start with enzymes. By definition, they are biological catalysts that speed up chemical reactions. And so, what do I mean about chemical reactions? Well, when you're eating food, the food is not going to just break down on its own. It needs a bunch of different types of enzymes to break down all of these complex molecules into smaller bits and pieces that your body can use. But what's neat about an enzyme is that it doesn't get used up in the process. It's sort of like free labor. The enzyme is more than happy to do the work of breaking down or building up molecules, and it doesn't need ATP or any type of energy to do it. And so you can ask, okay, what would happen if we didn't have enzymes? What would happen if there were no enzymes to break down the molecules? Well, first of all, we wouldn't be alive without enzymes, because if we left it up to physics to break down all of the molecules for us without enzymes, and what do I mean by leaving it to physics? Well, for example, like if you leave a piece of bread on the table, it'll take months. You can leave it there for months and you'll see a lot of mold. You'll see it like slowly break down into bits and pieces, but it won't completely disappear. It won't be down to its smallest unit. And that's because physics alone can't just break down molecules. It takes a long, long time. It takes years. But with enzymes around, you can speed up that process, and that's why it's called a biological catalyst. That's why it said that enzymes speed up reactions, because now they don't have to rely on physics. Now they have the ability to break down a lot of different types of molecules that are necessary for life, and at a quick rate. We said enzymes are biological catalysts. They speed up reactions. And here's an example. So I don't know if you're lactose intolerant, or if you know somebody who's lactose intolerant, or if you know what it is. Lactose intolerance is the inability to digest milk. And milk has this molecule called lactose. And so lactose. And lactose needs to be broken down to create glucose and galactose. And then your body's cells are very happy to interact and gain energy out of these smaller units, okay? Your body's cells can't do anything with lactose, like they can't digest this molecule, but if you break it down, they can digest these. Now, a person who is lactose intolerant, that means that they don't produce the enzyme called lactase. And so lactose can only be broken down by this enzyme called lactase, and if it is, then you get these nice byproducts here. You get these very usable byproducts that your cells can use. Now, for a person who is lactose intolerant, they're not making this enzyme called lactase, and that means that the lactose goes straight into the digestive system, and then the bacteria in your gut are the ones that end up breaking down lactose, and they create all kinds of gas in your gut, and it makes you go to the bathroom. And so this is just one example of how important even just one enzyme is to our functions. And there are different types of enzymes. For example, when you eat protein, well, there has to be something to break it down, because having this whole molecule of protein is not useful, but having every single bit of amino acids is useful, because now we can rearrange these, or cells can rearrange these and make different types of proteins. And so the example for the enzyme that breaks down protein molecules is protease, okay? And you can find this in your stomach, this molecule, this enzyme here. And there are also amylases. These are enzymes that are in your mouth, ready to break down starch and glycogen. So it breaks down complex carbohydrates into smaller bits and pieces called glucose. And glucose can be easily used in your mitochondria to create a bunch of ATP, to create a bunch of energy. And enzymes don't just work alone. Mostly they work in a chemical reaction. They work with a bunch of other enzymes in a series of events. So let's say when you have glucose here, glucose requires a whole bunch of enzymes before it gets broken down to its smallest components of ATP, or before it's being used to make ATP. And so all I'm trying to say with this picture is that enzymes don't necessarily work alone. All right, let's move on to the cell membrane. We already mentioned in our previous lecture that the cell membrane is the protective part of the cell. It regulates what goes in and out of the cell. But now we're going to learn this extra concept of cell membranes, which is semi-permeable. Semi-permeable means that only certain things can go in and out of the cell, not just anything can go through it. And so you have to ask yourself, okay, why is it important for not just anything to go through the cell? Well, if you want to protect the cell, just like you want to protect your house, you don't want intruders in there, then you got to regulate what goes in and out of it. We mentioned earlier that the cell membrane is made up of these fatty phospholipids, and so that means that it has a phosphate group and a lipid tail, and that gives it a hydrophobic property. So what does hydrophobic mean? Phobic means phobia. I am scared of hydro. I am scared of water, meaning that these nonpolar tails do not interact well with water. They repel them, just like magnets repelling. And so you can imagine having a fatty layer around your cells. You know that most things are going to be repelled away from it because nonpolar doesn't like interacting with polar stuff. So it's very good at being protective. Now the cell membrane is also made up of proteins that are embedded inside of them. And we mentioned earlier that proteins have many functions. Many functions. They are the building blocks of life. And some of these proteins can act like channels, which allow only certain molecules to go in and out of the cell, especially if those molecules are hydrophilic or polar, meaning that if those molecules have positive and negative charges like water, and we know that molecules can be easily repelled by the fats, well, why not have a tunnel or a different means to get through the cell without having that issue of being repelled? And so some proteins act like protein channels. Others act like antennas, receiving signals from the environment. Others act like proteins to just maintain the structure of the phospholipid layer. And so they have many functions. So let's talk about the 6 major functions performed by proteins. And you should know this in case I quiz you on it or test you. And so the 6 major functions of proteins embedded in a cell membrane, right, are this. So they help maintain the cell shape so that it doesn't just fall apart. It helps with signal transduction, meaning that a protein can deliver a message into the cell to perform a function. So it can work like a messenger. It also performs enzyme activity. So maybe some molecules need to be broken down outside of the cell or inside of the cell before they pass through. It helps with cell-to-cell recognition. And so this is how cells can recognize each other. Okay? So that they can know what type of cells they're interacting with, and that's how tissues are made. And it helps with transport, meaning that it can move molecules in and out of the cell, especially those molecules that are going to be easily repelled by fats. And that's what gives it its selective permeability, meaning that it's very selective in what goes in and out of the cell. And it also helps with intracellular junctions, meaning that it helps cells fuse together, right? So if they fuse together, then they become tissues. So here's just a picture of them. We said that it's helpful in transport and enzymatic activity and signal transduction, meaning that it can just simply interact with the outer environment and send a signal to the cell, and that tells the cell to do something. Either it'll make a protein or it'll protect the cell. It can serve, the signal can be many different things. It helps with cell-to-cell recognition, so that's how cells recognize each other, and they even eventually can fuse together by intracellular joining. And it also helps attach to the cytoskeleton. But I want you to remember the list that I gave you right before this slide. Okay, so that's the list that you need to know. We said that cells have selective permeability, meaning that it just doesn't let anything pass through. And so, again, this is the cell membrane here made up of lipids. Lipids are nonpolar. They don't like interacting with molecules that have charges. So let's take a look at this chart. Out of all these molecules here, so we have nonpolar, meaning that there's no net charge, and polar, that it has net charge. This one doesn't have a net charge. This one has a net charge, doesn't, and it doesn't. Out of all these molecules, which ones do you think will easily pass through the cell membrane? So remember, the cell membrane is nonpolar, and it can easily interact with other molecules that are nonpolar. All right, so I'll let you think about that for a second. And just real quick, the cell membrane is like a barrier with a semi-permeable membrane. So like these little, like pretend like this gate has pores and only things that are tiny enough can pass through, especially that are nonpolar. But for the most part, things are kept out of the cell. All right, so if you said that oxygen can easily diffuse through the membrane because it's nonpolar, you were correct. Carbon dioxide can easily go through the cell membrane. Ethanol can easily go through the cell membrane. Vitamins can go through the cell membrane. And even tiny molecules, we said that water is polar, but if the molecule is tiny enough, like water, only some will go through without totally getting repelled, but it has to be really tiny. But for the most part, water is transported through channel proteins like aquaporins, and that allows it to easily go in and out of the cell. And also glucose and other polar type of molecules need assistance to go through the membrane. And so why do we even care about this? Well, like I had mentioned in the previous lecture, that drugs themselves are molecules, they're macromolecules. And there's quite a bit of them that are nonpolar, like THC, for example. So THC is a component found in marijuana, and that is a nonpolar molecule, and so it can easily diffuse through your cell membranes, and that's how it has an effect on you, on your cells. And so is ethanol. Ethanol is another nonpolar molecule, and we know that ethanol can easily diffuse to the cells, and that's what gives a person sort of like this feeling of or a change in feeling or change in the way that they're functioning, and that's because ethanol is basically entering the cells and causing these changes. All right, so we just already went over nonpolar and polar. So we said that anything that's water soluble, which is polar molecules, cannot easily transport across the membrane. They need assistance. But anything that is lipid soluble, like nonpolar molecules, can easily transport across without the help of a protein channel. And some examples of that are steroids, ethanol, and vitamins. All right, let's talk about the types of transport. So I'm talking about the transport of molecules and substances going into and out of the cell. And there are many ways to go into and out of the cell. And we're going to talk about that. So some of these methods don't require energy, meaning that they don't need ATP. They can just easily pass through simple diffusion, sort of like those nonpolar molecules that we talked about. Or they need some assistance, or they can pass through osmosis, and we'll talk about that in a minute. And some transportation needs ATP, and this is called active transport, endocytosis, and exocytosis. And so if I ask you which types of transport need energy, then you would answer active transport, endocytosis, exocytosis. And if I ask you what type of transport does not require energy, then you would answer passive transport, facilitated diffusion, and osmosis. Passive transport is also known as simple diffusion. Let's start off with simple diffusion, which is also called passive transport, so you can use either one to refer to the same thing, which is basically the movement of molecules from high concentration to a lower concentration, provided that there's a semi-permeable membrane. And just to give you a quick example of diffusion, like a general answer or a general example of diffusion, think about what happens when you spray air freshener on one corner of the room. And so you release these molecules of air freshener into one corner. And eventually those molecules will diffuse, they'll spread throughout the room because the room walls are its limitation. And so it'll spread throughout the room until it reaches an equilibrium, until there's an equality of molecules all over your room. And the same thing happens with passive transport. Basically, so let's say the yellow lines are the cell membrane, and there's some type of molecule outside, let's say sodium, that wants to get inside of the cell. It can easily do it without a protein channel, and depending on what kind of property that molecule is, but it can do it as long as there's a high concentration of that molecule on the outside, which causes it to enter into the cell. It's a driving force until it reaches equilibrium on both sides. And so cells just happen to take advantage of that free energy. That is basically a law of physics, diffusion, and doesn't really need to, it's kind of like free labor. It doesn't need ATP to do this. So simple diffusion is the random movement of particles down a concentration gradient going from high to low, means that it does not require energy. And some of these examples are like small molecules, non-polar ones such as oxygen. Oxygen is very much needed in your cells. I mean, without oxygen, we wouldn't be alive. And so it goes into your cells very easily just by this act of simple diffusion, going from high concentration of oxygen in your blood to inside of the cell, which is lower in oxygen concentration. And of course, there are drug molecules that take advantage of this pathway, like ethanol. Facilitated diffusion means that there is some assistance. So it's a type of passive transport that does not require energy. And so let's say it's a protein. But in this case, this protein channel is pretty much always open. It just easily allows molecules to enter from high concentration to low concentration. For example, like water molecules. Water molecules need these proteins called aquaporins, because that's how it easily gets into the cell, and it takes advantage of the fact that there's more water outside of the cell than inside of the cell. And so since there's more outside, it needs to spread into the inside until it reaches an equilibrium. And so that's facilitated. Facilitated means that the protein itself, like the protein channel, allows for this movement of molecules that are not liked by the hydrophobic layers. Remember, fats do not like polar molecules, and so they need this protein here to allow it to easily diffuse in and out. Osmosis is specifically the diffusion of water. So anytime I mention anything about water, I'm referring to osmosis. Just like, I guess, what I mentioned in the previous slide, right, with the aquaporins. But in this case, nothing's different other than the same idea that water is moving from high concentration to low concentration. And so whenever you hear the word osmosis, you must think water and the movement of water, because water can also go from the inside of the cell to the outside of the cell, depending on what's going on. And so just know that cells also take advantage of osmosis just to be able to function in everyday life. And it's literally the diffusion of water through a selectively permeable membrane. We already know what that means. Going from high concentration to low concentration through a passive transport. Now, active transport means that there needs to be some type of ATP, some type of energy. It's not like a door that easily opens or a window and you can just get into the house. This time you need a lock to open the door. So you need extra energy, extra effort to cause this change. And so active transport can move substances in and out of the cell through against its concentration gradient, meaning that even if there's a lower amount on the outside than there is on the inside, it can force the opposite of physics. It can force molecules to enter the cell, even if there's a high concentration inside, because there's extra energy that is helping this process to occur. So this right here is a protein channel, and this protein channel has been opened and closed by energy, by ATP, which allows molecules to enter in and out of the cell, even against a concentration gradient. So it doesn't matter if the concentration is high on the outside or if it's low on the outside. That's irrelevant. this extra energy by used by ATP allows this movement of molecules to easily enter in and out of the cell. And so I'm sure you can imagine that molecules need to be on both sides of the cell for many reasons. We can't just always depend on high concentration to low concentration. We need to be able to easily maneuver molecules in and out of the cell. And so that's what active transport does. So again, active transport requires energy. Here's an example of active transport. No molecules will pass through unless ATP opens up that channel. Whereas of passive transport, and we said that the passive requires no energy, would be diffusion into and out of the cells, and facilitated diffusion, just a protein channel that is always open, but it's going against a concentration gradient. It's going from high to low, high to low. Whereas here, whereas this example of active transport, we go from low to high. Okay, we go against a concentration gradient. All right, let's switch to a different type of transport, and this is the last type of transport, yet the most popular types of transport to get big molecules into the cell, especially different types of drug molecules, and this is called endocytosis and exocytosis. Endo, first of all, means particles from the outside of the environment going into the cell, and so the cell membrane basically fuses around these particles. It creates a vesicle and it protects them, and now they can enter the cell. So endo means going into the cell. Whereas exocytosis, these are more examples of endocytosis. So like big proteins that need to get into the cell that are too big for protein channels, they can go into endocytosis. And these are just different types of endocytosis, but don't worry about them. This is just examples for you to look at. All right, now let's talk about the opposite of endocytosis. So that is exocytosis, and these are big molecules that can't just easily go through diffusion or easily go through a protein channel, that need to be transported outside of the cell. And so vesicles are also formed around them, and then this vesicle fuses onto the membrane, just like you see here, and then releases the chemicals into the environment. And so this is a major mode of transportation for the molecules in your brain, the neurotransmitters. So I don't know if you've heard of neurotransmitters, but those are the main molecules that cause many functions for your neurons to fire like really quickly or slow down on their firing and whatnot. And so this is a major mode of transportation, especially in your neurons. Okay, and so that's how drugs also get in and out of the cells. And so if I ask you which mode of transportation allows huge molecules like big proteins and neurotransmitters to exit the cell, you would say exocytosis. And what if those big molecules want to enter the cell? Well, you would say endocytosis. And this right here is an actual real image through an electron microscope showing exocytosis. And so these molecules are exiting the cell. And like I mentioned, your neuron, so this is a neuron right here, uses this to allow neurotransmitters to go into and out of the synapses. All right, and just a quick picture, exocytosis leaving the cell, endocytosis entering the cell.